Thanks, Remy. That's a great presentation. You know, the slides you show around pace of development, it's just remarkable how much exploding activity there is. And I think you highlighted very clearly that we don't have the tools. We don't have the clinical tools. We, at this point, have enormous amounts of data that are being generated and don't really have a way of trying to work out what it all means. So there may be things we already should know that we don't know. And the clinical tools, even clinical trials, we just don't have the tools where we could fine-tune and try and avoid the failures that we actually see with large trials. And be able to select something prior to that would obviously be a huge step forward. I would just make one comment around investigator-initiated trials, just to remind you that the approval of pembrolizumab in MSI-high was based on an investigator-initiated trial. So there is significant value in that area. So thanks, Ramy. We're going to move on now to Roy Herbst. Roy, who is well known to all of us, Ensign Professor of Medicine and Professor of Pharmacology, and Chief of Medical Oncology and Director of the Thoracic Oncology Research Program, and Associate Director for Translational Research at Yale. And Roy has obviously spent his entire academic career actually focusing in lung cancer, and I think he's going to use lung cancer as the platform or the substrate to talk us through the current state of monotherapy and the clinical trial status for the combinations. So over to you, Roy.
Thank you, Roger, and thank you to the committee for inviting me today. And I was asked to talk about the limits of monotherapy, and actually I'm set up very well from the last talk. So what I thought I'd do is talk first about lung cancer as an example, reviewing the promise and limitations of immunotherapy. Then a little bit about the mechanisms of sensitivity and resistance to immunotherapy, primary and acquired, and I'll show you some of our own work from Yale. And then combination immunotherapy, principles and practice, a few examples from lung cancer, but not too many details, really focusing more on the policy issues. And then really, how are we going to personalize immunotherapy for patients, which really I think is a theme of this conference. Well, lung cancer's had great progress. It's amazing what's happened in 20 years, from chemotherapy, docetaxel, platinum alone, and then, of course, anti-angiogenesis agents with a great deal of promise, never quite reached what we hoped it would as far as the results. But then, of course, all the targeted therapies. And it really has been all about targeted therapy in lung cancer for the last 10 years. These agents work. We're now finding molecular drivers of 1% targets. But, of course, no one's ever cured with those agents. Resistance consistently develops. And now, in recent years, it's really immunotherapy. And immunotherapy has taken the other 85% of patients who don't have a driver, an addicted driver, and allowed them to get therapy. So how well is it working? Well, it's working well. I wanted to show this as an example here. I know there are some advocates in the group. This is Maureen. She's a patient of Scott Gettinger. When I was looking at a job at Yale 8 years ago, Scott and Mario Snow showed me these films. And you can see this is a woman with squamous lung cancer, 3 prior therapies, who came in for a phase 1 trial, that first phase 1 trial that's published in the New England Journal of Medicine, and had an amazing response within months, in the lung and the liver. Actually stopped drug at 2 years arbitrarily. That's the way the trials were designed. And she's doing well 8 years later. No therapy for the last 6 years, perfectly healthy, perfectly functional. Is she cured? That's something we can discuss. It's getting pretty close. The question is, how common is her outcome? I often ask this. I'm going to be in Vail in a few weeks for the course where we work with the students on clinical trials, and I use this slide. How common is this? We have the commercials on TV. Everyone comes into the clinic. The patients expect they're all going to have this outcome. It's only 10 to 15%. So with all the trials, with all that Rami just showed us, with all that's going on, it's still only 10% or so that are having this amazing result. And it's great to see exceptional responder-type projects, but there's a lot of work still to do. Acquired resistance occurs in more than 50% of patients, even if they have stable disease or response. And I'm focusing on lung cancer, but it's pretty consistent with other tumors as well. There will be a recurrence. And then there's much more room for improvement. Well, you saw this slide. In fact, I think it was the very same slide. We must have gotten it from the same place. I'll just focus on a different aspect. These drugs are approved, but look at this. These curves are all coming down to the bottom. we got a lot of work to do. We need to personalize this therapy. Some of these drugs were approved without a biomarker. This drug here, pembrolizumab, with a biomarker. Atezolizumab had a biomarker. It worked in both biomarker negative and positive, but we need to do better. Most of these patients will still die of lung cancer even though they're getting immunotherapy. I don't think that's really known out there by patients, certainly, and many physicians don't realize that yet as well, and caregivers. Now, personalization is important. I'm not going to talk too much about this because we have a whole session later, but biomarker PD-L1 makes sense. We know that we're targeting this, so there should be PD-L1 in the tumor. This is an assay that was developed, either no PD-L1 staining, this is membranous staining of PD-L1, or more than 50% of the cells positive, and then an in-between group. And in a very nice phase 1 trial that Roger and his group were involved with, they actually saw that in those patients who had the high PD-L1, their survival was better than those who had 0 or low. So the marker did have some predictive ability. It's not perfect, and you'll hear about all the issues with it later in today's meeting. But when you use that now in the frontline setting, here's data that Julie Bramer recently presented, Keynote 24. This is now frontline therapy for lung cancer with pembrolizumab, using that PD-L1 and taking those patients that have more than 50% of their tumor positive. So now when you use the marker, you can get survival curves like this, where the median survival is 30 months. 20 years ago, the median survival would be 6 to 8 months. So amazing progress. There is a tail to this curve. We're now at 3 years, and the data are going strong. This is the control group that got chemotherapy. There was some crossover, but clearly it's better to get the drug first in the right patients. So it's having great benefit, but again, can we raise this tail even higher? And then, of course, just to show you that it's not so simple, these are data from the ASCO meeting just a month ago. This is another trial that's looking at the same drug, but now we're looking at patients who have greater than 1% PD-L1, and you can see the results are good, but not quite as good. The hazard ratio here is only 0.81. It was 0.69 in the slide that I just showed you. But then I guess I didn't put that slide in, but if you look at the patients who have PD-L1 in the 1 to 49% group, there's very little benefit. So what's my point? You need predictive markers. We need to know who to treat, who not to treat. What about sensitivity and resistance to these agents? Well, we know it's a complex immune microenvironment. We're talking today mostly about PD-1, PD-L1 for this conference, as was agreed upon, but there's a whole bunch of other things going on in the immune microenvironment, regulatory cells, helper cells, macrophages. So it's really not going to be simply you just block this. There are going to be resistance mechanisms either on target or off target. What we've done at Yale, and we're very influenced, of course, by Li-Ping Chen, who's part of our group in our lung spore, and this is work from David Rim, is we took 450 cases of lung cancer, archival cases, not treated with PD-1 or PD-L1 inhibitors. And what we did is we asked, what's the status of PD-L1 and TIL, tumor-infiltrating lymphocytes? I would contest that if you're on target, if you have the ability to respond to these agents, you probably need both. You need to have PD-L1 in the tumor, and you need to have TIL. So the TIL is blue, and the PD-L1 is brown. And what we found, and this has been confirmed now by many other groups, is 17% of patients had that. These are probably the patients that are benefiting from the therapy. But then 26% of these patients were what we call type 3. There's plenty of TIL, plenty of blue, but there's no PD-L1. So if this is true, and I think that these results can be believed a little bit more than some of the patient samples that we ordinarily get, because these are whole tissue samples, not just core biopsies, that would mean there are other checkpoints involved. So we shouldn't be giving these expensive drugs. We shouldn't be giving these drugs up front. We should be thinking, what are the other checkpoints involved, and using those. And then the most important group, and I'll get to this in a second, is the group type 1 and type 4. There's no TIL. There are no tumor-infiltrating lymphocytes. The most common group of patients with lung cancer, you can see, have no tumor-infiltrating lymphocytes and no PD-L1. So these patients aren't going to benefit from this therapy. This is a non-inflamed tumor. And here's a group that has some PD-L1 and no TIL cells. This is probably oncogene-derived. So the type 2, just focusing on that, we just recently trying to define what is resistance is going to be the first step as we target it. And I bet if I went around the room, everyone would have a slightly different definition of it. The way we're thinking of it right now is the type 2 patients here, these are the patients that have the target. They have the TIL, they have the PD-L1, and this is a group where there is either going to be response or no response. If there's no response at all, that's primary resistance. That's probably the 50% patients with pembrolizumab. Half of those patients don't respond. There's a primary resistance here. We need to figure out what's going on. But But then many of those patients will respond, and they'll either be a durable responder or they'll acquire resistance. And again, what's going on there? We need biopsies, we need tissue, as we just heard from Rami, to figure that out. And then there are all the other groups where they don't have the targets, and there are other things going on, and there are other reasons why they're not going to respond to these PD-1, PD-L1 agents. Well, this slide helps to explain a little bit about that. This is from the first phase 1 trial of a drug that was called MPDL3280, now atezolizumab. We actually did this in very close collaboration with Dan Shen and Ira Melman, a group of investigators from around the world, where basically we took patients and we treated them with this drug, and you can see wonderful response here in the lung. This is an example of a patient who's responding extremely well to an immune checkpoint inhibitor. And with biopsies pre and post, as we just heard about, if you look at 0 and 4 weeks, you can see the CD8 cells increasing. And I think it would be great if we could do this with imaging and not have to biopsy the patient, because I can tell you, having done some of these, the patients, it's hard for patients to do these biopsies on study. Then you can not only look at the T cells, we know that we have the cytotoxic T cells, but we can also look at the quality of the T cells. So it's not just are there T cells, but are they active? Can they generate the immune response? And here you can see an immune chip where you can actually look at RNA transcription, and you can see pre and post, pre in green and post in yellow. And here's granzyme A and B goes up. Perforin, actually making a little hole in the tumor cell to kill it. All these things go up. This is an active immune response. This is not going to help us predictively, but it tells us what we're looking for so that we can understand why it doesn't work and how we can influence it in better ways. Now, here are real data from that trial. Here are 3 examples of patients who did not respond. And here you can see the pre-treatment biopsy and the on-treatment biopsy shown here. And you can see here's what we were calling immune ignorance. These are patients where you don't see any CD8 positive cells pre and none post. I don't show you the immune chip results because there is no change. They're both down. Here's a non-functional immune response. You're getting a little bit of activity, but if you look at these T cells, there's no evidence of increase in those enzymes that I mentioned. And then this is very interesting, this sort of excluded infiltrate, where the T cells come to the tumor, but they don't get in. They don't get to the tumor to generate the adaptive immune response. And all these are examples of patients where if we understood that this was happening in advance, perhaps we could influence it with the right combination. And this is actually a slide that I borrowed from Dan Shen. This is the way, I think, based on those data and other data, one way to think of this is that you might have a tumor that's inflamed, a tumor that's immune excluded, or the immune desert, the immune ignorant tumor. And all these, knowing in advance, and we'll hear about that throughout the 2 days, we can think about specific therapies, not just giving the same therapy to all. We've been doing work, and this is work from Kurt Schauper, David Rim, and our lung spore group, and David might show some similar slides. But if we're going to look at T cells, we need to be more quantitative about how we look at T cells. So here are two different patients with lung cancer, and you could look at this and say, well, there are some more T cells here than here. Call this 1+, call this 2+, or 3+. We've all read slides like that in the past. But now if you use quantitative immunofluorescence with multiple fluors, now you can actually see that CD3. You can actually see the CD3 positive cells. And if you look at a series of patients, you can see there's a large variability, the red being the CD3 positive cells. These are 2 series, one from Yale, one from Greece, our colleague Konstantin Sirangos in Greece. And you can see now we can quantitatively measure CD3 cells. We can do a lot of other things, but for today I'm just showing you the CD3. Then we can take our 4 groups, and then we can try to sort of be a little bit more specific as to how we quantitate. So we have the 2 groups that are low CD3. Those are the immune desert. Those are the patients with the non-inflamed tumors. Then we can sort of find 2 more groups, and this is just our first iteration of this, where you can look for high CD3, but either low granzyme and low Ki67. So the CD3, the T cells, are not really active, or you can look at those cells that have high granzyme and high Ki67, and you can see they assort like this. So this is now work in press, and I'll just show you the top-line result on the next slide. Basically, if you start to look at these data and you correlate this with outcome, and we've been collecting samples for 8 years at Yale, now, of course, on label we're collecting as well, you can see that, again, it's early data, but this green line here, these are the patients who have group 2. Actually, the group with low granzyme and low Ki67, not how we initially thought this would work out, but our current theory is that these are cells that are exhausted and waiting for the priming from the PD-1 or the PD-L1 agent. This needs to be confirmed in larger studies, and we're doing that through collaboration. Another theme of this meeting, this is the Stand Up for Cancer Lung Group, now in its 4th year, about to start its 4th year. all these sites working together. We have clinical trials, but we also have a commercial use project where patients who are getting these immunotherapies on label are now getting the drugs. We're getting the tumor, we're sequencing, that's being done at the Broad, and we're doing the immune profiling at Yale with the panel shown here. And of course, here's the panel that will look at the granzyme and the Ki-67, so hopefully more on this in years to come. Then, of course, what about acquired resistance? This is work that we've done led by Katie Paletti and her team, working with Rick Lifton before he left for Rockefeller. We said, what if we sequence patients pre and post? And we were very fortunate. We've had a rebiopsy program in place for many years. So you can take patients who are pre-treatment from immunotherapy, they get the immunotherapy and they respond, and then they become resistant. A very valuable set of patients, very hard to find this. And even when you do find these patients, you then look and much of your tissue has gone off to Genentech, Merck, or whoever you're working with on the clinical trial because you need a lot of tissue to get patients on study. But we've been collecting it, and then you can analyze it. Patient has their own control, and you can look at the pre and post biopsies. And we actually did that. The one drawback here is the drugs that were used were either PD-1, PD-L1, or a combination of PD-1, PD-L1, and CTLA-4, so a mixed group. This is published in Cancer Discovery for anyone who wants to see it in more detail. But you can see 14 patients. We had 28, but it turned out we had 14 good pairs. You can see the green is where the patients responded. The response to these agents in lung cancer tends to occur early. Here's one late responder. The green triangle is when they became resistant. And lo and behold, when we looked at this, we had, for example, in this one patient, patient, response, and then resistance. And actually, when we looked at this, we actually found that the patient actually, in the diagnostic specimen, already was missing one copy of the beta-2 microglobulin gene, but on resistance had lost both copies. So interesting result because, of course, as most of you know, beta-2 microglobulin is an important component of MHC1. So here's a tumor that's lost the ability to present antigen. Of course, here you could use all the PD-1, PD-L1 checkpoint inhibitors in the world. It's not going to work. We need other approaches that don't require the antigen presentation. So those are just a few examples. I could talk all day, but it's very exciting as we figure out mechanism. We get biopsies. We understand why are patients resistant and treat them in the right way. Combination therapy. This is my version of I borrowed this from Mark Phillips. You can see Matt Phillips, many trials. Rami showed this so well. This is from Dan Chen and Ira Melman. Many different combinations that we could use. In fact, this is just a list of some of the PD-1, PD-L1 combinations in lung cancer. The list is large. In fact, it led Gina Kolata to write an article for the New York Times, A Cancer Conundrum, Too Many Drug Trials, Too Few Patients. I don't think there are too many trials. There just aren't enough of the right trials and the scientifically guided trials. In fact, you can make the case that having these trials is good because it allows patients who don't have resources to get these drugs. And actually, this paper highlighted a woman with breast cancer who is getting therapy on trial who didn't have insurance that would have paid for it. Now, there are many different mechanisms, and I'm not going to talk about these today. I would just contest that you would want to think about why a cell is resistant, whether it's the microenvironment, whether it's the lack of the target, and treat them in a specific way. You'll hear from the group this afternoon or a little bit later about CTLA-4, PD-1, PD-L1 combos. Those are looking quite promising in lung cancer. Tumor mutational burden has been used as a marker there. We'll hear about that later, but that's something to keep in mind as the meeting goes on. We'll hear a bit more about the microenvironment today as well. We already heard a bit from Rami. There are many studies ongoing here. We've done one that we've led out of Yale, looking at pembrolizumab, the PD-1 inhibitor, plus a vascular endothelial growth factor inhibitor. The idea being that this has an effect on dendritic cell presentation and also on the microenvironment and allowing the T cells to better get to the tumor. This was a trial that was done in lung cancer, GI cancers, and several others. And just showing you that you can see the results, that here's a single phase 1 trial with very nice results in lung cancer, showing responses even in patients who are PD-L1 negative. I actually show this as a pro and a con. I think this is very exciting, but I wouldn't believe a minute of it until it has a phase 2 comparison against a control, and that's hopefully ongoing. The data for time to progression in refractory lung cancer, IO naive, was about 10 months. But again, many studies like this, but they need correlative studies and they need to be confirmed. So then who would have thought chemotherapy combinations would be the first one out the block? Certainly not me. The fact that we're using chemotherapy, potentially killing the very effector cells that we're trying to promote. What's the effect on memory cells? Who knows? Many, I'm not going to read through this slide for the sake of time, but many reasons why we probably need to think about this quite carefully. How much chemotherapy, what sequence of chemotherapy. Nonetheless, it does seem to work. Long-term effects on the memory cells and on durability of the response is something that's going to need to be followed. But here are the data. Rami showed the same slide, and again, I'll point out the same thing. Looks great. A hazard ratio of 0.49. I sat on the ASCO Meaningful Outcomes Committee a few years ago, in fact, shared it for the lung cancer group. We said 0.75 would have been a meaningful outcome. 0.49, it's amazing. But still, look at the time to progression curves. 68% of patients are still progressing at 1 year. So it's good, but there's a lot of room for improvement. But it's perfect. We have drugs that work. Now we can actually figure it out through the science, and hopefully that will be the case in the next few years. Finally, what is the next step? And just as I conclude, I think we need to personalize immunotherapy. That's what I've tried to show you with these few vignettes. I'm not going to go through any of this except to say that we need to look at all this, and we have this today. We need to look at tumor mutational burden, liquid biopsies, microbiome imaging. It was great to see that in the first talk. Some of the work that I've mentioned, RNA profiles, TMB, sequencing. All this is going to have to be put together, of course, in a composite model. We're doing studies, and as are many, we need studies with tissue. So here's a study of nivolumab, ipilimumab in lung cancer. It's already in phase 3. The results are pending. Some of them have now been presented. But this is a study that we're doing with multiple sites where we have the laboratory group involved through our lung spore, where we're getting tumor tissue, blood, and stool from microbiome in all patients, and really throwing the kitchen sink at it. We need more studies like this to really figure out who responds and who doesn't. It's that database that Rami mentioned, pulling it all together. The lung master protocol, just a word about that. We'll hear about master protocols later today. We've now taken this study that has over 1,700 patients, and we're really focusing it on IO refractory patients. In lung cancer, we've had a hard time with this trial because the landscape has changed so quickly as we've done it over the last 5 years. But now almost everyone we see is going to have refractory disease to immunotherapy. So we're now focusing on combinations that might work in the refractory setting. And the beauty of this trial is through the public-private partnership, everyone's getting tissue. So tissue correlating with outcome. So what I've tried to show you, it's been a journey. 10 years ago, it was targeted therapy for lung cancer. In fact, we've been working on that for almost 20 years. It's 20 years since the first studies with EGFR inhibitors were done with lung cancer. It took 4 or 5 years before the biomarker was determined. John Mendelsohn stood at this podium many times and made that case. And now for immunotherapy, biomarkers aren't just involving the tumor anymore. Now it's involving the microenvironment as well. So we're working on it. And I would just say that now the next steps are to bring these drugs to other stages of disease, including earlier disease, explore activity in other types of cancer, biomarkers and science to better determine mechanisms of sensitivity, and to really personalize immunotherapy. What we need to do, and few are doing it, it's very hard, get the tumor, biopsy the tumor, and use those results to develop the right combination. It will happen. We've spent over 20 years developing personalized therapies for targeted therapy. Now the same must be done for IO, and it's even more complex. But I think with groups like this, we'll be able to do it. Thank you very much.